Which one is... Okay, great. Welcome to today's Immunology seminar. It was my absolute pleasure to introduce our speaker, Dr. Leyuan Ma, who is currently a faculty at Penn. Leyuan received his PhD at UMass Worcester under the mentorship of Michael Green. Leyuan was extremely productive and published the 3 first author research articles addressing drug resistance in chronic myeloid leukemia. After that, Leyuan moved 50 miles east and did a postdoc with Daryl Irvine in MIT. There, he designed the amph-FITC-CAR T ligands that prime CAR T activation in lymph node and triggered massive CAR T expansion, increased the donor cell polyfunctionality, and enhanced the anti-tumor efficacy in multiple immune-competent mouse tumor models. And this is an exceptionally innovative approach to boost CAR Ts, especially targeting solid tumors. And this work was published in Science. In 2022, Leyuan joined the Department of Pathology and Laboratory Medicine at the Penn Medicine as an assistant professor. His lab currently focuses on immune engineering and employs a combination of genetic, chemistry, engineering, and computational tools to decode the molecular and cellular crosstalk between immune cells and their microenvironment, and leverages these crosstalk mechanisms to engineer novel biomaterials, proteins, and cell-based precision immunotherapies. I think Leyuan will tell you more details in a second. Not surprisingly, because of his astonishing scientific contributions, Leyuan was awarded with multiple prestigious awards, which include the American Cancer Society Postdoc Fellowship, NIAID New Innovator Award DP2, W.W. Smith Charitable Trust Award, Melanoma Research Alliance Young Investigator Award, and the Ben and Catherine Ivy Foundation Award. Without further ado, Leyuan, the floor is yours.
Hey, thank you so much, Xiaolei, for the introduction. And of course, I really appreciate this opportunity to share my work here to the broad audience. So I guess everybody can hear me well. So I will start talking. Okay, great. So today I'm mostly gonna share the work that I've been accomplished since my postdoc, and this include 2 stories. The first story is essentially setting up this platform, this synthetic vaccine platform. The second story is we kind of followed up an unexpected phenomenon called antigen spreading, essentially only triggered kind of when we combine the synthetic vaccine with CAR T cell therapy. That essentially is able to deal with this a major issue in solid tumor therapy, kind of using CAR T, which is kind of antigen heterogeneity. Okay, so I'm sure kind of in this audience, kind of probably I don't have to introduce too much about the immune system or T cell, but I still want to touch kind of very briefly on this. So kind of within solid tumor, there are multiple different kind of immune cells within the microenvironment, including macrophage, NK cell, dendritic cell, different type of T cells, and neutrophils. So among these immune cells, macrophage and neutrophil are mostly kind of pro-tumor. And NK cell, T cells are kind of anti-tumor. Dendritic cells are mostly there kind of amplify the immune response. So in order to lead to very effective tumor destruction, you need a balance of the number and also the functionality of these different type of immune cells. So among these immune cells, CD8 T cells are essentially the foot soldier of this that can directly interact with tumor cell and kill them by releasing a variety of different effector molecules such as granzyme, porphyrin, and interferon gamma. So this is just one of the cartoon kind of showing the potential combination immunotherapy that is highly effective. And however, once you deplete CD8 T cells, now you almost completely lose this kind of immunotherapy effect. And this really shows the importance of CD8 T cell in this kind of immunotherapy. So T cells can recognize tumor by recognizing through their T cell receptor, recognizing a special complex which is called peptide matrix complex. So this peptide matrix complex is comprised of a little epitope, a linear small peptide epitope that derives from intracellular proteins. And then this interaction is highly, highly specific and very sensitive, and it can recognize as low as 1 molecule. And also kind of, because this kind of a discovery that T cell can recognize the antigen highly specifically and highly sensitively, people have been using basically, cloning out this TCR gene and then putting that into the non-tumor reactive T cells. And as long as the TCR is tumor reactive, now you can turn a population of non-tumor reactive T cell into a tumor reactive T cell. But the problem is that if you're just using TCR, there's a phenomenon called MHC restriction and MHC polymorphism. So that means, every human individual, they have 6 different MHC1 allele in each individual. And if you want one, for example, cancer-specific new antigen to be presented on that MHC1 and then recognized by one single TCR, it's really kind of, if you do the math, it's really kind of not that many patients can benefit from this kind of therapy because this MHC matched patients are very, very low. So therefore, these CAR T cells have been developed. So the CAR T cell was engineered in a way that by fusing, talking about the chimeric antigen receptor, it's kind of analogous to the TCR, but it's totally different. It's created by fusing the antibody antigen recognition domain to the intracellular domain from a T cell receptor and also the co-stimulation molecules. So now you have a chimeric kind of molecule, which essentially serves as an orthogonal sensor for the T cell to trigger T cell activation. So because it recognizes surface protein instead of the peptide present in complex with MHC, this makes them MHC independent. And this is kind of how we actually leverage this MHC independence to develop this synthetic vaccine, making it broadly applicable instead of the conventional vaccine, the only limited to certain a patient bearing certain MHC alleles. Okay, so far, the CAR T therapy has been super successful in the leukemia and lymphoma models, and this is just one of the early examples, showing the clinical trial result. Once you're injecting the CD19 CAR T cells to the patient bearing this acute lymphoblastic leukemia, and now in bone marrow, just within a month, you can see essentially all of the tumor cells got cleared out, and up to 80% patient can have this long-term survival. And right now, FDA have already approved 6 different CAR T products, including 4 CAR T products for B-cell ALL and lymphoma, and 2 for BCMA targeting BCMA for multiple myeloma. So, however, as you can see on the right, the CAR T therapy against solid tumor has not been very successful. So this was one of the clinical trials that carried out a couple of years ago at Penn. They were basically kind of leveraging a very unique, tumor specific. So I'm emphasizing tumor specific because it's very rare to identify such a sort of a mutated surface receptor protein that only expresses on tumor cells. So they are developing CAR T cell against EGFR-3, which is a highly specific tumor antigen on the cell surface. However, but as you can see, the efficacy is actually not that high. So initially, patients have some response, and eventually, most patients start to relapse, eventually succumb to the disease. And I'll explain later so why. So for now, I just want to briefly mention why we want to study this. So and solid tumor have been having many different type of solid tumor, and a lot of patients bearing solid tumor really does not benefit for this very potent therapy. So in order to understand, how we can improve the CAR T therapy for solid tumors, we need to understand the difference between the CAR T therapy within solid tumor versus leukemia models. So in leukemia setting, so CAR T therapy, the chimeric antigen receptor T cell infused into the patient essentially coexist in circulation with their target cell, including both the CD19 positive leukemic cell and also healthy B cells. So this coexisting in circulation and this kind of very frequent interaction essentially provide abundant antigen stimulation for the CAR T, promoting their early stage expansion and eventually leading to their long-term engraftment. And so this is highly critical. But for solid tumor, as you can see, most of the antigen is confined in this little area that is highly immunosuppressive, and the antigen is also very limited. And this limited antigen exposure, immunosuppressive microenvironment within solid tumor, eventually leads to limited CAR T expansion and reduced persistence and functionality, and finally leading to limited efficacy of overall efficacy of CAR T therapy against the solid tumor. There are, of course, many other hurdles, such as lack of tumor-specific surface antigen, or antigen loss, or tumor antigen heterogeneity. So this is one of the histology data showing the variable expression of EGFR-3 across different patients bearing glioblastoma. So that's kind of one of the major reasons why the EGFR-3 CAR T therapy against glioblastoma failed, because that's mostly because of the relapse of the antigen-negative tumor cell eventually cause the patient to lethal disease. Okay, so now we have been trying to to address this kind of CAR T persistence and long-term expansion issue. So basically, our goal is to develop a vaccine, because we know T cell naturally expand very potently to vaccine stimulation. So we are hypothesizing, why couldn't we develop such a vaccine for CAR T specifically? And we did like make a couple of discoveries here. So one is that we are able to using this vaccine to address the limited CAR T expansion, persistent functionality issue. And then, the second story I talk about will be how to leverage this vaccine to boost CAR T to potentially deal with the antigen loss or tumor escape because of this tumor antigen heterogeneity. Okay, so talking about the vaccine. So first of all, I want to briefly introduce like how the conventional vaccine works. So in the conventional vaccination setting, typically, you're going to formulate your vaccine by mixing your protein antigen with adjuvant. So the antigen is essentially providing the specific T cell response, and the adjuvant essentially will activate your immune system. Upon trafficking to the draining lymph node, the antigen and adjuvant will be captured by the lymph node resident antigen presenting cell. And then this protein antigen will be internalized and processed by within the endosome, eventually leading to the small epitope to be presented on the MHC and eventually presented to T cell through the peptide MHC TCR interaction. And this provides signal 1, which essentially defines the T cell response specificity. And then the adjuvant will activate DC cells, and that can upregulate co-stimulation and also promote DC to secrete this pro-inflammatory cytokine, which serve as signal 2 and signal 3. So only in the presence of these 3 signals, you will see the optimal T cell differentiation, proliferation, and memory formation. So this is highly critical for successful therapy using T cells. So now we want to borrow this concept to establish a synthetic vaccine for CAR T cells. So the CAR T cell recognize target through a totally different mechanism as we know so far. Like it recognizes surface antigen instead of recognizing something presented on the MHC molecule. So therefore, we need to specifically address this issue, how to put an antigen, like a synthetic antigen, on the surface of APCs or dendritic cells that can trigger CAR T activation within the lymph node. Because for the co-stimulation and pro-inflammatory cytokine, we can easily provide that by including an adjuvant. So to achieve this goal of putting a synthetic ligand on the APC in the lymph node, we basically elaborate a polymer we call amphiphile ligand polymer. This is essentially comprised of a lipid tail, which have the property of binding to serum albumin and also inserting onto the cell surface. And then we, in the middle, we basically put a solubilization linker there to help formulating this vaccine. And then on the end, we basically attach any kind of ligand recognized by the CAR, such as a small peptide, a small molecule, or maybe a small protein domain. And now, once you put in this lipid polymer, or we call it amphiphile ligand, in the aqueous solution, such as water or PBS, they're going to form a micelle. But once you inject this vaccine into mice subcutaneously, they will encounter serum albumin. So the serum albumin have very interesting property that it bears 6 different or 4 to 6 different lipid binding domains. So therefore, once in the presence of albumin, the micelle will be able to break apart into a single polymer and be carried into the draining lymph node. And within the draining lymph node, this polymer can come off the albumin and then directly jump onto the surface of APC by directly inserting into the lipid bilayer. Now you have this synthetic ligand directly presented by APC. So this is our theory, and the next couple slides I'm going to show you how we prove it. So, in order to track the trafficking of this molecule, we essentially used a fluorophore labeled amphiphile ligand. We call it amph-FITC, and then we inject it into the tail base of the animal, and then we track their accumulation in the lymph node organs. Basically, you can see the signal significantly increased in the draining inguinal lymph node and distal exterior lymph node. And then we isolate the cells within the lymph node that are labeled by this amph-FITC molecule, and you can see all the signal localized on cell surface. This essentially proves our point. So once this molecule traffics into the lymph node, first, they're going to highly localize in lymph node. Second, they can insert onto the cell surface. And these cells, and we eventually figure out these are the professional antigen presenting cells. So this is how we figure this out. So, we, in order to know where this amphiphile molecule actually localized, on a subtissue level, and we basically did a tissue sectioning of the lymph node and then perform confocal imaging. So we stain this lymph node section with anti-CD3, D220, and anti-FITC to define where T cell zone, the general structure of the lymph node, such as T cell zone, B cell zone, and where this FITC molecule are localized. So as you can see from the imaging, most of the FITC signal is localized within the interfollicular region, where the most of the professional antigen presenting cells are localized naturally. So we also, dissociate the lymph node into single cell dimension, and then perform flow cytometry to define like which individual population actually carry this amphiphile FITC molecule. And as you can see, most of the FITC molecules are really localized within these professional APCs, including macrophage, medullary macrophage, and C11C, C11B double positive DCs. And also the FITC, because we are also using an anti-FITC antibody to check if the FITC still remain on the cell surface. As you can see, there's still decent percentage of the cells bearing FITC on their cell surface. So since we know like the FITC molecule, the amphiphile ligand can trap the lymph node and insert on the cell surface, now we want to see whether this concept of vaccination can really work in terms of boosting CAR T cells. So to establish this platform, we essentially used a anti-FITC CAR T by feeding the anti-FITC SCFV to the CD28 intracellular domain and CD3 zeta intracellular domain. And we confirmed this CAR have a very good, kind of expression on the mouse T cells. And for the amphiphile molecule, we confirmed it can also very efficiently insert on the cell surface. So then we performed some in vitro assays, just a very simple in vitro assay, just confirm this system works at least like as we expected. So we basically can label the target cell with our amphiphile molecule, then stimulate FITC-CAR T in vitro. And as you can see, by increasing the labeling of the cell with amphiphile, you can see increased stimulation of the CAR T as monitored by the interferon gamma secretion, which is one of the major marker of T cell activation. So this result shows that we can use this amphiphile ligand to specifically and potently stimulate CAR T cells in vitro. Now, how does it work in vivo? So to achieve this goal, we essentially can took advantage of this congenic mouse T cell transplantation model. We took T cells from CD45.1 mouse and then making CAR T out of it, and then transfer that into CD45.2 recipient. Now, this way we can track T cell proliferation and expansion based on the surface marker like CD45.1. So because in most of CAR T therapy, clinically, they do need to lymphodeplete the mice to empty the space for the CAR T cell to expand and engraft. So we also kind of adopted this kind of therapeutic regimen here. So basically what we did here, we did lymphodepletion and then transfer CAR T cells and then give like 2 doses of vaccination. So as you can see here, like we do have included one control group, which including like 10 million CAR T, which is typically the maximum number of CAR T cells you will inject into a mouse. And then, we also inject a much lower dose, like only 50,000 CAR T. Then we give them like either amphiphile FITC ligand or amphiphile FITC ligand plus an adjuvant, which is typically an essential component of the conventional vaccine. So what you can see here is that if we just injecting 50,000 CAR T, you can barely detect them at all kind of in the periphery. But if you're just injecting 10 million CAR T, which is the maximum dose you give to the animal, you can see roughly peaks at 40%. And then slowly kind of decreases. And if you give them lower dose of CAR T, but give them amph-FITC, which is the green line here, you will see kind of transient expansion, and then even the second dose of vaccine does not help that much. It's not really it's kind of comparable to directly injecting 10 million CAR T, but at a later stage. But now if you include adjuvants, now you can see a dramatic difference here. So once including both adjuvant and the amphiphile ligand, even just with 50,000 CAR T cells, you can see much more pronounced expansion by day 14, like reaching almost 70%. This means with the complete vaccine, including amph-FITC and adjuvant, you can achieve roughly 200-fold expansion of the CAR T cells, and also this expansion of CAR T not just give you better, more number of CAR T, but also potentially changing their phenotype. So we basically also followed the CAR T phenotype, their memory phenotype, kind of by the day 30 time point. You can see once we including the adjuvant, you actually significantly increase the memory CAR T formation. So why does the adjuvant matter? So as I mentioned, this adjuvant can upregulate the signal 2 and signal 3 by increasing this co-stimulation expression and also triggering release of pro-inflammatory cytokine. So we're specifically interested in the co-stimulation because the co-stimulation has been used in the CAR T construction. So therefore, we specifically monitor all different kinds of co-stimulation molecule that express in the dendritic cells in the lymph node. So as you can see, by including the adjuvant, you can significantly upregulate a variety of different kind of the co-stimulation molecule that hopefully can provide additional co-stimulation to the CAR T cells in addition to the building co-stimulation within the CAR. So this is our kind of hypothesis. So this is actually the experiment we did to prove that. So the way we did this is essentially we gave the mice the CAR T cells and then followed by complete vaccine. At the same time, we block or not block this like different type of co-stimulation molecule. So as you can see here, once you block the co-stimulation within different cocktail of antibody, you significantly reduce the CAR T proliferation or expansion within the lymph node and also reduce their functionality. So this means just having the building co-stimulation is not enough to achieve like optimal expansion. But once you provide additional co-stimulation, more like a natural arrangement, like because T cell naturally being activated by APC. If you put in the CAR T cell in this context, you likely almost can guarantee to have optimal CAR T stimulation. And of course, the optimal formulation of the CAR T vaccine should comprise both the amphiphile ligand and an effective adjuvant. Okay, so finally, so we know the synthetic vaccine works well kind of through the CAR, but how well it actually works compared with conventional vaccine. So to do this, we essentially leverage the TCR transgenic T cells, which is like, here we use like PML T cell, which is expressing PML TCR recognizing GP100 antigen. And then we transduce this TCR transgenic T cell with the FITC-CAR. Now you can have a bispecific T cell that can recognize both the GP100 antigen and also the FITC antigen. So we can stimulate these CAR T cells with either amph-FITC vaccine or the conventional GP100 vaccine. So from the plot on the right, you can see like, in this adaptive cell transfer setting at least, either vaccine kind of works kind of comparably. Like you can always, like the first dose of vaccine, you can boost the CAR T expansion up to 70, 80%. And the second dose later, kind of by day 40 time point, you can boost it again. And also by 70 times, by the 70, you can, the third dose of vaccine, you can also kind of boost it again. And so on the iris imaging, this actually only 48 hours after the first dose of vaccine. And both vaccines you can see just trigger massive CAR T expansion. And this CAR T we use actually have the luciferase. This is how you can actually monitor in real time how the CAR T expands in response to the vaccine stimulation. Okay, so now till this moment, I have been talking about this model vaccine and a model CAR T, but how this strategy actually works for a bona fide tumor targeting CAR, if we can generate such an amphiphile ligand to directly stimulate a anti-tumor CAR T cell. So to do this, we picked a model, which is also this EGFR variant 3 tumor model. And this EGFR variant 3 is the oncogene that's actually derived from a missplicing event that eventually create a very specific tumor-specific epitope called PEP3. And there are actually available monoclonal antibody that you can use to generate the CAR. And then basically building on this information, we were able to create a amphiphile PEP3 ligand kind of a vaccine. And then using this monoclonal antibody sequence, we can create this anti-EGFR variant 3 CAR T cell. And then by kind of doing this in vitro experiment using the amphiphile PEP3 labeled cells to trigger EGFR CAR T activation, we were able to confirm this CAR T cell very specifically responds to this vaccine. Now this is in vitro. Then let's talk about in vivo work. So this in this model we kind of established the tumor in the mouse first. And then we did lymphodepletion, then followed by CAR T transfer. Then we gave the mice only single dose of vaccination. Then we continuously sample peripheral blood to monitor the CAR T expansion in response to the single dose of vaccination. So as you can see, even the mice now bear solid tumor. But if you just inject CAR T, you do not actually see much of the expansion in the periphery. But once you give them single dose of vaccination, you can see CAR T directly bump up to more than 30%. And this effect can actually last to more than 3 weeks. And also we sample the peripheral blood to monitor the T cell phenotype. And as you can see with the vaccine stimulation, the T cell just have increased cytokine secretion, increased metabolism, and increased cytotoxicity. So based on this data, we are sure that the vaccine not just give you more CAR T cells, but also give you better CAR T cells. So that was like in the peripheral blood. But how about the CAR T cell within the tumor? So we so in a separate experiment, what we have done is that we basically like analyze the CAR T cell within the tumor. And as you can see with the vaccine boosting, more than 10-fold CAR T cells enter the tumor. And these tumor-infiltrating CAR T cells also exhibited increased proliferation and also polyfunctionality, as monitored by this KRC7 staining, different kind of granzyme B cytokine markers. And also we performed like bulk RNA sequencing to compare the CAR T cell within the tumor from vaccine boosted or non-boosted mice. As you can see, like there are actually significant increase of the proliferation signature as shown here, the MYC target and E2F target, which is kind of consistent with our KRAS-7 marker staining. And the rest of the signature is very interesting. Almost all of them kind of are localized in these metabolic pathways, like such as oxidative phosphorylation, mTOR signaling, fatty acid metabolism, or peroxisome signaling. So that means this vaccine boosting can be one of the major effects of vaccine boosting can be metabolic reprogramming. Therefore, it can enhance CAR T functionality. So this basically shows like the vaccine boosting, that's the final outcome of the vaccine boosted CAR T therapy. Without a vaccine boosting, the CAR T only mildly like slows down tumor growth, but not leading to long-term survival. But by including the vaccine, you can actually significantly slow down tumor growth and actually cure roughly 60% animals. So till this point, what you can see that the CAR T the vaccine increased CAR T number and also increased the functionality. And this actually almost a twofold effect. So in the next experiment, we try to see like if the qualitative change of the CAR T cells is sufficient to enhance their anti-tumor efficacy. So the way we did this is we did a sequential transplantation of the CAR T cells. So first of all, we transfer CAR T cells into donor mice, and that does not bear tumor. So this way, then we kind of vaccine boosting them or using controlled PBS to boost them. And then after 7 days, we isolate this boosted or non-boosted CAR T, then transfer them into a secondary recipient, which kind of bears solid tumor, and then without additional treatment. So this way, you can because we are transferring equal number of them, we can really specifically control the T cells and to monitor if the qualitative change of the CAR T cells that caused by the vaccine boosting can really lead to any difference. This is the result. And this we definitely say, this the CAR T cells have significantly increased functionality on the per cell basis, because when the tumor binding mass receive equal number of CAR T cells, the vaccine boosted one significantly delayed tumor growth and also even cure more mice compared to the one that's non-boosted. Okay, so the immunotherapy is quite different from the conventional small molecule-based therapy in that it can trigger memory formation that's potentially lasting, causing this long-term therapeutic effect. So in the CAR T therapy setting, one of the memory effect will be the memory of the CAR T cells. So that means if you challenge this cured animal with the antigen positive tumor, likely they're gonna get rejected. So this is exactly what we saw. So in the naive mice, which never received CAR T therapy, all of the tumors start growing. But in the cured mice, all of the mice reject this antigen positive tumor. So then we did a second experiment, challenged the mice with antigen negative tumor. And very interestingly, we also say all of the tumor cells got rejected. So apparently, because the CAR T cell cannot recognize this antigen negative tumor, so the rejection of the tumor must be caused by the endogenous immunity. This actually was achieved through a process called antigen spreading. So during this antigen spreading, CAR T cell will be trafficking to the tumor site, start killing tumor cells, and then the tumor debris will be captured by this migratory dendritic cell, which can carry the tumor antigen to the adrenal lymph node, where they're going to present the antigen to the endogenous T cells. Now, once the endogenous T cells recognize tumor antigen, they're going to get activated and amplify, eventually traffic out of the from the lymph node and back to the tumor site. Now, this is almost the second wave of the army coming over here to dealing with residual tumor cells. This is actually what we have found so far. So we did a variety of different combination of our vaccine without the adjuvant or without CAR T, and eventually found that only with the complete vaccine formulation, which means including amphiphile ligand and adjuvant, then plus CAR T, you can really trigger this massive antigen spreading. But once you're missing one component or just with CAR T alone, you really don't see much of the antigen spreading. Okay, so now we know this endogenous T cell response can be triggered, but does it have any significant consequence? So in order to do this, we essentially perform the same vaccine boosted CAR T therapy in either wild type mice or regular knockout mice. So in the wild type mice, they will have this immune system. They have T cell. You can monitor the antigen spreading there. But in regular knockout mice, they do not have endogenous T cells or B cells. Therefore, there's no antigen spread. So what you can see here is that in the wild type mice group, when they receive the CAR T plus vaccine therapy, you can see most of the tumors they regress and they never came back. But in the regular knockout mice, although kind of within a certain period of time, the tumor kind of start regressing and to a level that's undetectable, but slowly kind of most of them kind of rebound, eventually kind of relapse. And we analyze the tumor using flow cytometry and monitor the antigen expression, as you can see on the plot on the right. Kind of most of the relapse tumor, they always have significantly reduced surface antigen expression. That means the CAR T cell cannot recognize them very efficiently anymore. And also kind of this can be the reason why in the wild type mice, this antigen spreading is really critical for dealing with the residual tumor cells through a totally different mechanism, like through the TCR recognizing antigen present on the MHC to getting rid of this residual tumor, preventing tumor relapse. So now we know this, this endogenous T cell response or antigen spreading is able to deal with residual tumor, preventing tumor relapse. But how can this phenomenon actually be harnessed to directly treat a tumor with pre-existing antigen heterogeneity, such as this EGFR-V3 positive glioblastoma? So in the patient, so as a diagnosis, they already have kind of various level EGFR-3 expression. So definitely it's the loss of EGFR-3 is not kind of really happening at the end of the tumor, but rather kind of at the very beginning. So whether we can leverage this vaccine boosting of a CAR T therapy to directly treat these kind of heterogeneous solid tumors. So the way to model this is that we are artificially kind of mixing the antigen negative and antigen positive mouse CD2A glioblastoma cells and creating like a mixed tumor, kind of having a range of antigen negative tumor cells there. And then we implant this tumor into either wild type mice or reg1 knockout mice. So as you can see in the reg1 knockout mice, the therapy does not really work as long as there's even the minimal amount of antigen negative tumor cells. But in the wild type mice, even up to 50% of the antigen negative tumor cells, this vaccine boosted CAR T therapy can still cure decent number of the mice. But of course, you can see the trend, as long as you increase the amount of antigen negative tumor cell, the efficacy of this vaccine boosted CAR T therapy also drops significantly. But through this experiment, we are 100% sure that this vaccine boosted CAR T therapy plus the antigen spreading triggered by this therapy can efficiently deal with the antigenically heterogeneous tumors. Okay, so now we know this vaccine boosting plus CAR T therapy have a very strong outcome, but we also want to understand the immunological mechanism behind there. We did a kind of series of studies, and eventually we came out to this conclusion that the interferon gamma really plays a central role in triggering antigen spreading. So as you can see here, when we induce this antigen spreading using the vaccine boosted CAR T therapy, but in block different cytokines, by blocking TNF alpha, you have no effect, but when blocking interferon gamma, you almost completely lost this antigen spreading. And we also confirmed this phenomenon by using interferon gamma deficient T cell to produce the CAR T. And this result also showed that the CAR T derived interferon gamma actually serves as a trigger for the antigen spreading. And then we perform a time course study, essentially blocking interferon gamma at different time points. And as you can see, only the early stage blockade actually have the strongest effect. When blocking interferon gamma at later stage, you really don't completely neutralize this antigen spreading. So this means CAR T produced interferon gamma actually played a major role in triggering antigen spreading at the very early stage. And then once you block interferon gamma using this genetic knockout in the CAR T cells, you almost completely lost the therapeutic efficacy of this vaccine-boosted CAR T therapy. And then we perform single-cell RNA sequencing to profile the tumor-infiltrating endogenous T cells, essentially as a way to monitor in the functional level, so how CAR T vaccine therapy actually can engage endogenous immunity, triggering antigen spreading and altering their phenotypes. So as you can see from the experimental setup, we also use this congenital transplantation model. We transferring CD45.1 CAR T to CD45.2 recipient. And then after 2 rounds of vaccination, we isolate this tumor-infiltrating endogenous T cell, then perform this, so we call it sequel, but it's also kind of essentially a single cell sequencing technology. We are able to recover roughly 10,000 non-CAR T cells and then phenotyping them on day 7 or day 14. And as you can see, the CD8 endogenous T cells got significantly expanded. And the Tregs in the vaccine boosted group actually significantly reduced. And we also kind of characterize their phenotypes. So interestingly, in the CD8 compartment, you can see like significant increase of the cytotoxicity gene signature. But in the CD4 T helper cells, you can see a shifting of the T cell phenotype from Th17 to Th1. And then we kind of focused on the P15E specific T cells. This is kind of public epitope, kind of recognizing endogenous retroviral antigen. So basically, we also found that with the vaccine boosting, these retroviral specific T cells have been significantly increased their cytotoxicity. And all the rest of the cell with unknown specificity have the similar phenotype. So this single cell RNA sequencing analysis essentially confirmed our finding that the vaccine boosting not just increasing the number, but also shifting the T cell phenotype toward the more of a anti-tumor phenotype. Okay, so then we did a series of ELISpot and also kind of using genetic knockout and antibody blockade, eventually trying to reveal the potential mechanism of a CAR T therapy-induced antigen spreading. So here, we already discovered interferon gamma plays a central role here, but then we know that these C cells is supposedly also play a central role in promoting the antigen spreading by triggering endogenous T cell response. So therefore, we use the beta-3 knockout mice, which is essentially missing the cross-presenting DC, which is the central DC population that are responsible for presenting the antigen to triggering endogenous T cell response. And then we kind of knock out this interferon gamma receptor within the recipient mice, which also kind of almost abolishes completely the antigen spreading. Or if we're knocking out interferon gamma receptor on the DC cell surface specifically, you also see significantly reduce the antigen spreading. So this kind of ties up pretty well like how the interferon gamma actually affects the endogenous immunity. And then we also know that interferon gamma can potentially act back on the CAR T cells as an autocrine mechanism. And as you can see here, once we knock out interferon gamma receptor on the CAR T cells, we also completely lose antigen spreading. So this means the interferon gamma not just acting kind of on the endogenous immunity, but also acts back on the CAR T cell to form a positive feedback loop. So we also know from previous work that DC can also secrete IL-12, and the IL-12 can also act on CAR T cells, maintain their enhanced cytotoxicity, and therefore we performed antibody blockade to neutralize IL-12. And you can see this almost phenocopies like the interferon gamma blockade and almost completely blocks antigen spreading. And also we kind of knocked out IL-12 component P40 from the recipient mice, and this also kind of neutralized antigen spreading. And finally, because based on our previous study, we know metabolism played a major role in CAR T cells, and once they receive kind of the vaccine boosting, therefore we hypothesize this if we block this metabolism related gene, the PGC1 alpha, we can also see reduced antigen spreading, and this is exactly what we saw. So basically, kind of once you knock out PGC1 alpha, you see reduced interferon gamma secretion and also see reduced antigen spreading. So now this is essentially the final mechanism we have figured out. The CAR T produce interferon gamma or increased amount of interferon gamma in response to vaccine boosting. Then interferon gamma acts on the DC and also acts back on the CAR T cell. So once interferon gamma acts on the DC, it can promote DC maturation or activation, which can secrete IL-12, which can further act on CAR T cells to maintain their functionality. So since we know this interferon gamma plays a major role, to trigger antigen spreading, now we ask whether we can, genetically engineer the CAR T cells in a way that allow the CAR T cell to produce increased amount of interferon gamma in response to vaccine stimulation or antigen activation. The way we did this, we essentially putting the interferon gamma under control a amph-FITC promoter and then putting that in the CAR T cell. So this way, only when CAR T receives the stimulation, they can trigger interferon gamma secretion. Therefore, they can increase the total amount of the interferon gamma being secreted. So in vitro, we have confirmed that by introducing this genetic circuit, you can see almost increase of the interferon gamma to twice as much as the wild type CAR T cells. And also by introducing this these new CAR T cells into the mice, even without a vaccine boosting, you can still see increased antigen spreading compared to the wild type CAR T. And then once you include a vaccine boosting, you can further increase the antigen spread. And then we basically eventually test this new CAR T therapy in the mice bearing mixed tumor. So here we strategically picked like the tumor bearing 20% antigen, which we know previously just by vaccine plus CAR T therapy, we can cure roughly 50%. Now we have a model that we can like can evaluate if increasing interferon gamma secretion, we can further boost the therapeutic efficacy. And also this is exactly what we saw. So with CAR T plus vaccine boosting, you see roughly 50%, and with without a vaccine boosting, but just giving CAR T more interferon gamma, and you can cure roughly 3 out of 10 mice. And then when you combine both, you actually see significantly increased cure here. Okay, so this is the summary. Till now, we have developed a amphiphile synthetic vaccine. So we call it amphiphile vaccine, which essentially comprises the amphiphile ligand connected to, the CAR ligand connected to lipid polymer. And then this can hitchhike a serum albumin, which can be carried into the renal lymph node. And once they enter the renal lymph node, and this amphiphile ligand can jump onto the APC cell surface and be presented to the CAR T cells. Together with co-stimulation and pre-inflammatory cytokine. And this vaccine stimulation essentially significantly increased CAR T functionality, increase the number, and then CAR T can traffic to the lymph node, to the tumor site to attack tumor, and eventually leading to tumor death and the tumor antigen release. The tumor antigen can be captured by dendritic cell, which can traffic to the renal lymph node, eventually activate the endogenous T cells, and the activated endogenous T cell can come back to the tumor site to continue attacking the residual tumor cells. And the mechanism why this is essentially triggered through the CAR T producing interferon gamma, and interferon gamma can act on DC, DC can secrete IL-12, and through this positive feedback loop, we are able to, using the vaccine to sustain the activity of CAR T and also endogenous immunity, and eventually maintaining the long-term tumor control, and even leveraging this platform to directly treat tumors bearing heterogeneous tumors. Okay, so this is my acknowledgement, and this is my current team, and this is my funding source, and we have postdoc positions available immediately. Any candidate who are interested, feel free to reach out to me.